Okay, maybe for a professor, you can even select who is the best students in your lab. What is the best fit, just based on the genome profile. Okay, it's just like online shopping. I don't know how frequent you guys do online shopping. Sometimes they will suggest, hey, once you buy this, the screen will jump something, okay? And you can buy this based on the pattern you use, okay? Maybe it's interesting to buy this kind of stuff. Similar idea. Once you know what type of genome, what kind of person, the basis he has, maybe he tend to buy something like this, or maybe he tend to behave like this way. This is going to be a very big impact to our society. Okay. Oh, so right now they have a project called 1,000 genome running around the world. It's not just running on one or two individuals. That's called 1,000 genome. Actually, they have over 1,000 individuals are being enrolled. Okay. Those individuals are sampling from different populations. Some from America, and some from Africa, some from Europe, and some from Asia. Okay. So basically, they want to study whether among those populations there is a significant between that. Okay. You may wonder, hey, who is going to be sequenced next? Who will be sequenced next? One big prediction is that's you. Why did I say that? When I show you the price of sequencing, that's about 5-10 years ago. The price is only a country can do that. And in 8 years ago, or 5 years ago, still very expensive. I can't afford. But right now, when you want to complete a genome, it costs around $4,000 US. That means around 100,000 new Taiwan dollars. And in 5 years, the predictions is the cost of sequencing of a human genome is expected to go down to around $1,000 US. Much affordable. That's what I mean. That's only cost around 30,000 new Taiwanese dollars. This price is almost close to a new iPad. If you want to buy an iPad, you want to play around on the iPad. Yeah. Or you want to know your personal genome. You want to know what happened, what your genome can tell you. This is going to come very soon. In 5 years, in 10 years. This is going to be there. Okay. So what does bioinformatics really do? Those amount of data is very huge. 10 to the 9 nucleotides transfer into bytes is also at least 10 to the 9 bytes you need to use. Okay, this is computer terms. What that means, if you want to study 40 individuals, you will use at least around 1 terabyte this set of storage space to store the information. Not to mention, you need to have a very strong computing power to analyze the data. So you need a very very good technology to study that. And those kind of data, those kind of particular patterns rely on the computer to analyze. One of the things I want to show what has been done lately. Okay. So we want to take the medical information, with our genome information and our environmental information, to integrate those information together to study whether there exists a special pattern. Okay. So you may heard about there's environmental dangers in our society, but we are not aware of. For example, lately you heard about in the beef there are particular chemicals that may cause cancers. Okay, that's maybe the water we drink, the food we take, the things related to us that cause the disease happen, but we just didn't know. But with a long and detailed analysis, with those information all together, we have a way to find out that possibly cause. Even though probably at the beginning we have no idea what's happening. And one of the case I want to show is this has been done in the United States. And one of our projects we are doing with the Hilo Tangden Hospital is also we are doing similar project with that. Okay? But this project is extremely ambitious. Okay? So in this project, they studied about 100,000 individuals. So Kaiser Permanente actually is a very famous health provider in California, the Americans. They have a bunch of people registered in this health system. What does that mean? They have the histories of they going to the hospital. They have the histories about their medical histories. What kind of disease they have? Who do they talk to? What kind of doctor they talk to? What kind of medicine they took? What kind of environment? Where do they live? That kind of information they have. There are a bunch of information in their database. Most of the important, incredible things is they even collect 100,000 DNA samples. They sequence it using the technique what I'm talking about. Sequence their genes to analyze whether there's a potential difference or disease-causing genes in their genome composition. Using those informations, sorry, 2 minutes. Okay. Using those information, they want to study whether those things will lead to disease. Oh, so there's another case. Okay, I bet everybody heard about Google, right? You have done a Google before. One of the founder's wife, okay, Google's founder's wife, he founded this particular website. That's called 23andMe. Okay, 23 years does not mean the age. Okay, actually it means how many chromosomes, how many pairs of chromosomes in our body. Basically, they can take your saliva and run computer analysis, basically run your DNA analysis to figure out what kind of disease, what kind of risk you may have in your gene. Okay. This is done by Caucasians, and I believe one day it's going to apply to Asia. For Chinese, for Indonesia, for people living around. But one of the things I probably don't have enough time to talk about. Other than studying the disease gene, in our lab we are studying the disease antigens. We are trying to find out what will cause us to feel sick. For example, why some people, when the flu season comes, when some people are likely won't get sick, some people will get sick. We will study their background what those things can cause. Okay. I see the organizers keep telling me we should go faster. So that's okay. So one of the things I want to say is this is not only can apply to human genomes, we can also apply to different organisms. Okay. For example, that different fish that that has been sequenced. Okay, so actually I would like to end my talk right here. So one of the things I want to say is bioinformatics is very important features, and they need this is going to be a huge job market coming up. They need people to work on this, and this is a lot of business incentives to start with. And also, my lab is also recruiting people. If anybody is interested in bioinformatics, we need a good, hardworking graduate students. And okay, so thank you. So I take any questions.
Thank you very much, Mr. Sampon. The timekeeper told me to limit to only 3 questions for the last section. Okay, first question. Okay, from the back then.
Well, here, Professor, I'd like to ask you that what is the difference? Oh, sorry. My name is Sulida Fitri. I'm from Asia University. My department is Biomedical Informatics in Master. And my question is, what is the difference between DNA and the gene? Because is it DNA is a gene or gene is DNA? Some people are confused about it. And then, which one is more affecting in hereditary disease? Is it DNA or the gene? Thank you.
So the question is asking me the difference between the DNA and genes. Okay. So traditionally, okay, gene is composed with DNA. Okay. DNA is what we talk about in chemical terms, like AISC-Taiwan. Basically, there's four different elements in the DNAs. But gene is basically there's a long list of those DNAs who compose the genes. And gene itself, some of the genes can generate what they call the proteins. Okay. But some of the genes they cannot generate the protein. But usually we study the genes that can generate using the 2 important biological mechanisms. That's called transcription and translation. Basically, you transcribe DNA into RNA, and you use RNA to translate into protein. Okay. So whatever those genes can, whatever those DNA regions can do that's called a gene. That's called protein coding genes. Okay. But more specifically, some of the genes cannot generate to protein, but that's a different issue. Okay. So that's very good question, because the fundamental questions usually they will ask you to define what a gene is. Okay. Gene is usually we would like to define whatever the DNA factor that can translate into the proteins. Okay. Thank you. So you are asking whether the DNA and gene can so yeah, you heard my explanation. Basically, gene itself is a bunch of DNAs. So just using so DNA is the small elements of the genes. So when you study the human genome, usually you have what they call a chromosome. Okay, chromosome itself they contain many genes in different regions on the chromosome. But if you narrow the down a little bit, within the gene, there are a bunch of DNA inside it. Within the regions, there are a bunch of DNA. Okay. And so DNA is the basic element of the gene. Okay.
Okay.
Okay, let me introduce myself. My name is Logan. I am come from the National GIE University, and I am a master student of the department microbiology, immunology, and biopharmaceuticals. And I'm very interested about immunome. And you wrote on your PPT that immunome will be transformed 10 years later. That's right. But maybe you can explain more about that, because as I know that maybe I categorize self or non-self recognition about antibody. That's also maybe categorized about the immunome. So maybe you can explain more about the immunome or maybe the specific example or others. Thank you.
Okay, so one of the things I wanted to talk about is in my lab we are studying what is causing our immune system to respond. So this is also related to a concept that's called antigens. Basically, antigens usually considered as a bad stuff that invade our bodies. And our body can recognize those bad stuff and stimulate our immune system to fight against those bad stuff. Okay, so immunome basically is the study what kind of antigens, what kind of bad stuff will invade into our body, what will generate a good immune response. Okay. So in our lab right now, we are particularly study the self-direct response. We study what they call is T cell, CD4 plus T cell, to study what kind of antigens that will invade that may stimulate our immune response. This can be a good application to design vaccines. Basically, if you know what is a good antigens, for example, you know when flu season comes, you know what kind of flu is going to attack the human body. Okay, you can basically study what kind of segment can help you when you can give you a good vaccine. Basically, they can inject your vaccine to you when you're facing those flu virus. Okay, you will have the memory, and you can fight for that. You won't feel sick easily. And our goal is to find out a good sources that generate this kind of response, immune response. Okay, thank you.
Okay, from the right hand side.
Okay, first of all, I want to thank you. You guys are amazing. Okay, sitting here for so long. Okay, my students sit like 1 or 2 hours. They cannot take it. You guys sit here very quietly for 3-4 hours. I think that's amazing. I really do.
Thank you very much. It was such a sincerely honor for me to ask you, Professor, about these questions that I have been wondered. First of all, my name is Yuni. I am coming from Bogor Agricultural University, or IPB. Well, here I would like to ask you, as I heard from the various resources that instead of having a very positive impact, technology also has a very negative impact, such as gene mutilations. So I wonder, is it true or is it something about it, and also how that works out? And most important that I have been wondering about is how could we possibly dealing with that? Because according that most of us are technology users, including me, and I have nothing, I have no idea about that. So I would like to ask you about this one. Is that technology has a negative impact about the gene modulation and stuff? Thank you very much.
Thank you. So I can repeat the first question. So basically, you are asking whether the genetic alterations product affects us, whether have any good or bad effects to us, right? What is the second question? I didn't. Can you repeat the second question?
Well, my question basically is about, is it true that technology has negative impact about gene mutilations or DNA mutilations, and how that works out? Because, about the radiations, technology such as the computers or telephones, which has a really deep radiation. So is it anything to do with the gene or DNA gene mutilations, and how that works out, and how we could possibly deal with that problems? Thank you.
Thank you. Uni, right? Uni? Okay, thank you. Oh, those are very important questions and very important issues that impact our life. Okay, so one of the first questions, I'm going to give my personal opinion. Okay, I argue with my colleagues from time to time, so I heard positive and negative things. One of the things, as the question didn't state, whether what they call it, basically genetic modified products affect our health or our lives. Okay. The good thing we can see it very easily, but the bad things. People, what is the good things? Taiwan, 70% of the beans, the yellow beans, actually was imported from the from outside, was imported. Okay, they are genetically modified. I don't know if any guys have tried the traditional Chinese breakfast before. You drink? No? No. You just stay here long enough. Okay. One thing you should try is, okay, at least has a different taste. I don't know whether you will like it or not. That's what they call the bean milk. Soybean milk. Yeah, very good, soybean milk. The soybean itself, okay, mostly people use in Taiwan. Over 70%. That those are genetically modified products. Is it safe? This is not safe. Okay. As a scientist, what I can tell you is always have a hidden dangers inside this kind of material. Hidden danger, and what is hidden? Hide right there. We don't know yet. You change something, they didn't test long enough. You think how they test? The traditional soybeans, we have a test, like how long is human history? 2,000 years, 5,000 years, or over that. The soybeans, as we already know, is okay for our body, won't have a big effect. But probably not true for everyone, but for general population, the traditional soybean won't have a big effect. The genetic modified soybean, how many generations, how many years they have been tested? 10 years, 20 years? That's not long. A lot of things they just haven't seen yet. They haven't tried all kind of combination, right? If you do the experiment, we have to make a lot of different control just to see whether the specific factor will lead to particular effect. Okay. One of the things we don't know yet, whether this is real safe, whether that will cause, whether that will be a good, whether there's there's always a hidden danger inside the product. Okay. But one of the good thing we see, that's what everybody sees. Usually genetically modified product, what kind of benefit that they have? They can produce a lot. That's good for the farmer, for the merchants. Okay, they can sell it, get more profit. But whether that's good for our health, honestly speaking, I don't know. It can be very dangerous. It can be very dangerous.